Hello boys, welcome back to another session of video tutorials. This session is meant for class 11th biology students and this is meant for 26th August 2020. Our memory helps us in tracing out what we have been studying and linking the past understood material to the present one so that the total concept is clearly understood. We were talking yesterday in our session about mechanism of breathing, how exactly the lungs will inspire and expire. And in this session, we will be studying about the respiratory volumes and capacities. Now, what are these? Quantities of air the lungs can receive, hold and expel under different conditions. So this is called respiratory volumes. So in this we have got different set of volumes, and first one is the type of pulmonary volume or capacity is tidal volume. Pulmonary means lungs, okay? Pulmos means lungs. So first one is tidal volume. It is abbreviated as TV, not television of course, okay? Tidal volume. How to define that? Volume of air inspired or expired during a normal respiration, and there is no extra effort in this normal breathing. So that is tidal volume. So the value approximately is 500 ml. Quantity of air approximately 500 ml. Next, a healthy man can inspire or expire approximately 6,000 to 8,000 ml of air per minute. So next, IRV, inspiratory reserve volume, IRV. So it is additional volume of air a person can inspire, means inhale, by forcible inhalation. So there top TV, no effort, no extra effort. Here we are putting an extra effort. So this average is 2500 ml to 3000 ml in IRV, inspiratory reserve volume. So additional volume of air a person can inhale by forcible inhalation. Next, ERV, that is expiratory reserve volume. Expiratory reserve volume. Additional volume of air a person can expire by forcible expiration. Top IRV is forcible inspiration. ERV is forcible expiration. E for expiration. This averages around 1000 ml to 1100 ml. And next, RV, residual volume. So it is the volume of air remaining at the lungs even after a forcible expiration. This averages around 1100 ml to 1200 ml. Means how much is remaining the lungs. Residual means residue. By adding up few respiratory volumes described above, one can derive various pulmonary capacities which can be used in clinical diagnosis. So this is of great use in diagnosing particular disease in a person. So it helps a lot in clinical diagnosis. Now we come to IC, inspiratory capacity. It is the total volume of air that can be inhaled after a normal expiration. So IC is total volume of air a person can inhale after a normal exhalation. This includes tidal volume, inspiratory reserve volume. That means we can have a formula. IC is equal to TV plus IRV. So you can write down on a paper what is the formula. IC is equal to TV plus IRV. Next, EC. EC is expiratory capacity. So this is somewhere 1500 ml to 1600 ml approximately it should be, and IC should be 3000 to 3500 ml. This also you can note down. IC should be between 3000 to 3500 ml, and EC should be 1500 to 1600 ml. It is the total volume of air. What is EC? EC is expiratory capacity. It's the total volume of air that can be exhaled after normal inspiration. So how to write a formula? EC is equal to TV plus ERV. TV is tidal volume, ERV is expiratory reserve volume. So when we add these two, we get EC. And next we go to FRC. FRC is functional residual capacity. It is the volume of air that will remain in the lungs after a normal expiration. So FRC can be around 2100 ml to 2300 ml. So 2100 to 2300. So the formula can be FRC is equal to ERV plus RV. What is ERV? ERV already we said on top that it is expiratory reserve volume. So ERV plus RV. RV is residual volume. And then we come to vital capacity, VC. Vital capacity should be around 4000 to 4600 ml. And it is the total volume of air inspired and expired to a maximum level. So how can we write a formula for this? Since it is maximum volume of a person that can breathe in after a forced expiration. This includes ERV, TV and IRV are the maximum volume of air a person can breathe out after a forced inspiration. So here we are putting a lot of efforts in VC, vital capacity. So vital capacity is equal to TV plus IRV plus ERV. So that is how we can derive a equation. And now last, TLC, total lung capacity. It should be 5100 ml to 5800 ml. So 5100 to 5800 ml it should be. It is the total amount of air present in the lungs. It is the total amount of air present in the lungs and the respiratory passage after a maximum inspiration. So we can write the equation TLC is equal to VC plus RV. VC plus RV. VC vital capacity, RV residual volume. So based on these equations and formulae, we can have clinical analysis of a person. If somebody has got what normally doctors prescribe as RTI, respiratory tract infections. So during this RTIs, one can see these volumes, test for these volumes and analyze. Doctor will be in a position to analyze what is the exact problem with the respiratory system of the person. So here I want you to remember certain formulae. What is that? VC is equal to TV plus IRV plus ERV. Next, IC is equal to inspiratory capacity is equal to TV plus IRV. Next, EC. EC means expiratory capacity is equal to TV plus ERV. Next, FRC, functional residual capacity is equal to ERV plus RV. Finally, TLC, total lung capacity is equal to VC plus RV. So these are the formulae you must remember. Why? In examination he will give you some volumes or values in terms of the volume, and those you have to derive. For example, when I say VC is equal to TV plus IRV plus ERV, he will give you the volumes of TV, IRV, ERV. What is the VC? That is vital capacity. So you have to add all these values. Sometimes a substitution will be there. So you have to bring the VC this side and send that side. So these permutation combinations will be there, and finally you have to arrive at the solution. So some related to this, some problems, mathematical problems. I will post it in our group. You solve it and post the answers back. Then only I can understand that you have understood these tidal volumes. And now we go to the diagrammatic representation of exchange of gases at the alveolus and the body tissues with the blood and transport of oxygen and carbon dioxide. Now for this, what is that you have to understand is, all respiratory volume and capacities are about 20 to 25% less in women than in men. More in athletic people than in ascending, that is thin, non-exercising people, lazy fellows, I can say. With the exceptions of FRC and RV, all other respiratory volumes and respiratory capacities can be measured with the help of simple spirometer, an instrument called spirometer. With that we can measure. Now we come to exchange of gases. Now there are three layers here. One is the alveolus. In alveolus again, you can see here inspired air, it's entering into alveoli, and this is entering into pulmonary vein. You can see there marks, and pulmonary vein to systemic arteries. From systemic arteries, it is entering into body tissues. So oxygen is given to body tissues, but there is a condition. What is the condition? The partial pressure of oxygen should be maintained throughout. You see, when it is in alveolus, it is 104 mmHg, and when it comes down to the tissue level, the oxygen partial pressure, P means partial pressure. Partial pressure of oxygen is 95 mmHg. Okay, Hg refers to mercury, right? So from 104 in alveoli, when it comes to tissues, it has to be 95 mmHg. So this is in comparison to the atmospheric pressure, right? And then once the oxygen enters into tissues, hemoglobin combines with oxygen. Oxyhemoglobin will be formed. Normally we say pure blood is formed, and whatever digestive material is there, normally I said the end product of all the digestion is glucose. So this glucose combines with oxygen, 36 ATPs, Krebs cycle, remember, 36 ATPs are formed. Along with that H2O and CO2. H2O either will be utilized or it comes out of the body, and the CO2 now will be formed within the tissues. Our body cannot afford to keep this CO2 for a long time because it is poisonous. So when this is CO2 is formed in the tissues, it is 40 mmHg, partial pressure of CO2. Now from tissues it's coming back to systemic veins, particularly the long vein that takes all the impure blood from various parts of the body to heart. Now there it has to be 45 mmHg. Now from systemic veins carrying deoxygenated blood, from heart it comes to pulmonary artery, and pulmonary artery to lungs. And in the lungs, when it comes, the partial pressure of carbon dioxide has to be 40 mmHg, and then only it gets out of our body, expired air. So this is the main principle involved in exchange of gases. So breathing basically takes place with the condition that partial pressure of oxygen and carbon dioxide should be maintained. So alveoli are primary sites of exchange of gases. Exchange of gases also occur between blood and tissues, as you are seeing here. Oxygen and carbon dioxide are exchanged in these sites by simple diffusion method, and it is based on concentration gradient. Solubility of the gases and thickness of the membranes involved in diffusion are some of the important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure. That's what we are writing P. P refers to partial pressure. What is that? Pressure contributed by an individual gas in a mixture of gases. So when we breathe in, many times I ask this question: how is that when we breathe in, only oxygen is entering inside our body? In air, so many gases are there. When we breathe in through our nostrils, all the varieties of gases go in, but only why oxygen goes inside? Similarly, when so many gases are formed inside our body, how is that carbon dioxide is coming out? So the question is P, partial pressure. So partial pressure is to be maintained, and it is represented as you are seeing PO2 and PCO2. So partial pressure in the mmHg it is measured, and respiratory gas, if it is let's say oxygen, in atmospheric air it is 159 mmHg. In alveoli, as you are seeing, 104 mmHg. Deoxygenated blood 40 mmHg. Oxygenated blood, it has to be 95 mmHg as you are seeing, and in tissues it is 40 mmHg. So this partial pressure of these values are to be maintained, then only oxygen enters inside the tissues. Similarly, carbon dioxide. In atmospheric air, as all of you know, it is 0.3. Alveoli, it has to be 40 as you are seeing. And in deoxygenated blood, it has to be 45 mmHg. And in oxygenated blood, again it is 40. And in tissues, it is again 45 mmHg. So unless until this is maintained, the exchange of oxygen and carbon dioxide will not take place. Further, we will discuss in detail exactly within the alveoli how this partial pressure helps in exchange of gases in our next session. Until then, keep practicing, draw the